Good morning, everyone. I'm Simrita Costa, and on behalf of Altum Technologies, I'd like to welcome you all to our virtual webinar series on pharmacoinformatics. Our session for today is entitled as Computer Aided Approaches in Cutting Edge Drug Discovery. So, before we begin, I'd just like to give a very brief introduction to today's topic. Now, what exactly is computer aided drug design or CADD? So, this is a drug design approach that is technology driven, and since it's technology driven, it makes use of computational methods to design and optimize potential therapeutic compounds. It involves the use of computer models and simulations to predict how drug molecules will interact with biological targets like proteins or enzymes. So, computer aided drug design has revolutionized the landscape of pharmaceutical research and development. And what you're going to see today is the various computational methodologies that have been integrated into the drug discovery process. So, this includes molecular modeling, virtual screening, and QSAR, which is quantitative structure activity relationship models, etc. And I'd also like to add that this kind of an integrated approach to computer aided drug design can easily be done with BioVia Discovery Studio software because of the robust tools that it provides. And by leveraging these advanced tools, researchers can predict the interactions between drug candidates and biological targets. They can optimize lead compounds and streamline the drug development pipeline. And today's session will highlight the significant advancements and applications of computer aided drug design and demonstrate its crucial role in accelerating the discovery of novel therapeutics through a comprehensive analysis of current trends and innovations. So, let's understand more about this topic from our speaker of the day. It gives me immense pleasure to introduce Dr. June Moni Kalita. Dr. June Moni is an associate professor and HOD in the Department of Pharmaceutical Chemistry at Girij Ananda Chaudhary University, Guwahati, Assam. Dr. June Moni has done his B.Pharm from the University of North Bengal and was a gold medalist. He has done M.Pharm in pharmaceutical chemistry from Dibrugarh University, where he was again a gold medalist. Sir's research areas are computational chemistry, heterocyclic synthesis, peptide synthesis, and green synthesis. Dr. June has 4 extramural projects and 24 publications in well reputed journals. So, without taking much time, I'd like to hand over the platform to our expert. So, welcome, sir. And I invite you to take over the session.
Very good morning to all. Am I audible? Very good morning to all. Am I audible?
Yes,
Simran?
sir, you are
Yes, sir, you're
audible.
audible, sir.
Okay, fine. So, I'm Dr. Junmoni Kalita from Girijananda Chaudhary University, Assam, presently working as Associate Professor in Department of Pharmaceutical Chemistry. So, basically, I'm of pharmacy background, and so far I come to know that audience are from different backgrounds. So I kept the topic in general so that it becomes easy for everyone to understand the things. Okay. So the topic is computer-aided approaches in cutting-edge drug discovery. It's like it speaks about how computer-aided drug design helps in the process of finding new molecules to the market. So in general, what happens? A molecule to come into the market, it almost takes around 14 to 15 years, starting from laboratory work and again till that clinical trials. It takes around 14 to 15 years, and many places molecule they fails. So in those cases, computer-aided drug design helps, and it can reduce the rate of failure during clinical trials or preclinical trials, because many of the things we can predict uh, before going to the lab. That is the beauty of these computer drug design methodologies. Before going to the laboratory, before going to the real practical work, we can predict the activity, we can predict toxicity, even we can uh, predict drug-drug interactions also. So that is the beauty of computer drug design. So coming to the general part, what is drug design? What is drug discovery? Very synonymously, we use both the words, drug design, drug discovery. But both the words are having a bit difference. So drug means, as we already know, that any foreign molecule that enters our body, then if it can have any kind of effect or even effect also to our body, it is called as drug molecule. So good drugs are medicines, and some other drugs are there that is different. But whatever the medicines we are using, all are drugs. Okay. So now drug discovery and design. It is like discovery means it is kind of finding some new thing. At the same time, it is like means without any intention also sometimes we could find many drugs like penicillin, the best example, that is serendipity. Means this kind of things are called as discovery. But design is something else. Design means you know the target, you know your goal, and depending on the goal, you are designing something. That is drug design. So in case of drugs, what exactly happens is we know about the targets. We know about the targets. We know about the structure of the targets. And based on the structure of the target, that protein, we design our molecules. So it is called as design. Simply like if you have a piece of cloth and suppose you want to get a shirt designed for you, you will go to the tailor. Tailor will take different measurements and he will ask you about what kind of means style you want, short shirt, full shirt, half shirt, whatever specifications will be there. And based on those things, your shirt will be designed. So that is designing. So in case of drugs also, if we know about the targets, for example, suppose we all know that NSAIDs like aspirin, paracetamol, their target is COX enzyme, COX2 enzyme, cyclooxygenase 2 enzyme. So we know about the COX enzyme. We know the 3D structure of the enzyme. That enzyme is protein only. We know the amino acid sequences. We know the 3D structure of the enzymes. All these are already means available, and we even know about the active site structure. So when we know about the active site structure of the active site, then based on the active site, we can design our drugs so that it can bind there. It's like we know about the structure of the lock and we need to design the key. So once we know the structure of the lock, based on the structure of the lock, we'll be designing the key. So that is design. So that is not exactly discovery. So that is design, right? So when we know the specifications, what we are doing exactly means we know the target. So that is called as design. Next slide, please. So different methods of uh, drug design, drug discoveries were there. Like in eight, 1900th century, when aspirin it was discovered, perhaps in 1890 or 1894, like that. And surprisingly, aspirin it is one kind of drug longest running drug as well as having minimum side effect. So when it was discovered from a plant, Salix alba, and people in China, they found that when they took that bark of Salix alba, they got relief from pain. So they thought that something will be there in the bark of that plant. So they found that as to be salicylic acid. Later on, nowadays, it is salicylic acid is converted to uh, acetyl salicylic acid for other reasons. So now it is aspirin. So like that penicillin, it was discovered serendipitously by Alexander Fleming. He was doing something else. Suddenly he stopped at penicillin. So those kind of things were there. Then in 1990s, combinatorial chemistry came, where high throughput synthesis, high throughput screening was there. Means at a time, in short period of time, many molecules were synthesized. Then those molecules were screened. Then again, around 2000, new means computer-aided drug design era came, where all the things were done based on the targets now. So that is about modern era of drug design. So we'll be going one by one. Next slide. So this was how salicylic acid, it was discovered from Salix alba, right? So like this, many examples are there. Many medicines are from natural products. From natural product, even anti-cancer medicines, paclitaxel, docetaxel. So those are from natural products. Next. So this was salicylic acid. Later on, it was converted to aspirin. And based on the structure of salicylic acid, other things were also designed, like ibuprofen, with better efficacy and less toxicity. Other things were also later on synthesized. Okay. So this is about high throughput screening, where thousands of compounds can be screened for their activity in short period of time. That is around 1990, and those period it was means highly used. So coming to the computer-aided drug design part. Now, computer-aided drug design, it is a modern era of drug design, and its advantage is it can reduce the time, it can reduce the cost, and it can reduce the failure rate. This is the advantage. Means before getting into the real uh, practical lab or practical activity, we can do the things in the computer. We can simulate the things in the computer. And no doubt, it is not that easy to simulate a biological system. It is not that easy. But nowadays, the rate of success, it is increasing for computer drug design. So almost from 2010 onward, I'm using Discovery Studio. Before it was Accelerate Discovery Studio. Now it is BioVia. So I have seen from then onward, many of the modules are upgraded because day by day the programs are means new programs are added up, and day by day the computer system, it is getting advanced, and we are going to means we are getting more success rate in computer aided drug design. So basically, computer aided drug design, it can be of means many types. So four I have mentioned here: QSAR study, docking and virtual screening, Pharmaco modeling, and de novo drug design. Beyond this, we have homology modeling also there. Other things are also there. Okay. So QSAR study. QSAR means it is quantitative structure activity relationship. It is like whenever we will look into any uh, classification of drug, any classification of drug, will find like the drugs are somehow similar in their structures. If we'll go through NSAIDs like paracetamol, aspirin, ibuprofen, diclofenac, aceclofenac, if you go through their structures, you'll find their structures are somehow similar. They are similar. That is the reason they are acting on the same target, COX2. So they are similar means they are acting on the same target. So we need to find out what are the similarities between them. So that similarity between them, if we increase the similarity, we can increase the activity also. That is quantitative structure activity research. It is not only about the structural similarity, it is again physicochemical property similarities also will be there. Like lipophilicity is there, right? Other things are there. Okay. So based on the similarity, what is the relation with the activity that is measured? So activity it is quantified based on the similarity. Suppose at a particular position, if there will be electron withdrawing group, the activity is increasing. So that kind of similarity between the structure, it is found. Some other part, if some kind of lipophilic group is attached, the activity is suppose decreasing. That means what? In a new drug, we should not add any kind of lipophilic part at that particular position. So those kind of similarities are studied. And studying these similarities, equation is prepared. So based on that equation it is used to screen new molecules. So the concept is like we'll be taking certain number of drugs, means already means available molecules with their activity, and those molecules should have to act through the same receptor. Means if it is COX2, all the molecules has to act through COX2. Then only we can go for QSAR. So once those molecules are taken, so from those molecules with their activity, we get one equation like this. We get one equation like this, and this equation is having different kind of abbreviations like log P is there, that is lipophilicity. Okay, then sigma is there. Different things are there. So these are certain physicochemical properties. Okay. So once we get the equation, means this equation we got from the already active molecules. We got the equation. And now this equation will be used to screen new molecules. Suppose in one new library you have designed, you have 1,000 molecules and you want to means screen the molecules for their activity. So it is not possible to synthesize all those 1,000 molecules and get their activity done in the laboratory. It is possible, but it will take much time and much resources. So instead of going for that, you get one equation like this from the available molecules and screen the new molecules. And out of those 1,000 molecules, suppose you got 11 molecules, those are probably active. So for those 11 molecules only, now you will go for synthesis as well as practical activity. So that is the concept of QSR. Again, QSR, 2D QSR is there, 3D QSR is there. Okay, 2D QSR, simple kind of physicochemical properties like lipophilicity, electron donating substituents, electron withdrawing substituents, rough steric factor, those kind of properties will be considered. And in 3D QSR, these 2D QSA properties along with some other properties are also considered. Means three-dimensionally how the molecules is having different kind of electric field, like those who have done might be knowing about ComFine, ComSia. Okay, so those kind of analysis are done. So no doubt 3D QSA it is advanced form of QSA. So nowadays 3D QSA it is means used and it is having even a good success right now. Okay, next it is like next method is molecular docking. So in molecular docking, you know about the target. Okay. So previously in case of QSAR, you were not aware about the target. Already you knew about the molecules, available molecules. But now suppose you already know the target and even the target protein, it is available in PDB. Okay. So once the target protein it is available in the PDB, you know about the active site also. So easily you can take the molecule in the computer and you can go into the active site. You can check the how it looks like, right? And those newly designed molecules, you can check how it will bind with the target. If the binding energy it is fine, that means the molecule can be active in practical. Okay, so that is kind of thing it is. Where drugs are designed, drugs are taken into the active site, and in between many things are there. You need to energy minimize the molecules, even the protein. You need to clean the protein. Okay, many things are there. So finally, we get binding energy in the form of minus kilocalorie per joule. It is found in minus because it is delta G energy, Gibbs free energy. You you might be knowing about Gibbs free energy. That is, if it is negative, then the process is spontaneous. So it is like if it is negative, more is the negative, more the molecules are binding to the active site. It is like that. Okay. So again, after molecular looking, you can go for molecular dynamic simulation, where means means molecular docking. What we perform, it is kind of rigid thing. But in molecular dynamic simulation, each and every amino acid considered as uh, will be considered as flexible. So it takes much time and it is even deeper study. It is more detailed study. Okay. So it's like next slide. So it is like this structure of the lock, that is target. Then suppose key structure you have, means structure of the ligand molecule. Then it will be checked how it is binding with the uh, protein. So different amino acids will be there and different things are there like hydrogen bond donor, hydrogen bond acceptor, lipophilic site, uh, positive charge. So those things will be interacting with the amino acids wherever it is favorable. Means when it comes below 3 angstrom in distance, so it will bind there. So mainly this kind of means in the drugs, hydrogen bonds are predominant. Means between the ligand and the target, hydrogen bonds are predominant. Then other forces of interactions are also there, like van der Waals force. Then again, lipophilic interactions will be there, and very rarely covalent binding used to happen. Covalent bond very rarely used to happen. In very rare drugs are there. It's like in case of you might remember drugs, pantoprazole, omeprazole. Okay, so those proton pump inhibitors. These proton pump inhibitors they used to form covalent bonding with our proton pump, sodium potassium ATPase, what is in the stomach. Okay, so they used to form covalent bonding. But in case of molecular docking studies, we do not study about covalent bonding. We study about hydrogen bonding. electrostatic interactions, lipophilic interactions, van der Waals interactions. Okay, because we do not want drugs to permanently bind with the receptors or the targets. Because if you are taking one medicine, suppose beta blocker you were taking, beta 1 blocker, atenolol, those kind of thing, propanolol. So beta blocker generally what it does is it reduces the heart rate. So if the beta blocker, atenolol or propanolol, will permanently bind with the heart receptor, beta 1 receptor, it will permanently block the heart. So that we do not want. We want the drugs to be flexible, flexibly bind with the receptor. So majority of the drugs they used to bind flexibly with hydrogen bonding, with electrostatic interactions, with van der Waals interaction, and with lipophilic interactions. So it is flexible. It is totally dependent on the concentration of the drugs in the blood. More will be the concentration, binding will be more. Less will be the concentration, binding will be removed. It is like that. We do not want the drugs to bind permanently with the receptor. If it will bind permanently with the receptor, it will become poison. So that is the reason we only check for hydrogen bonding and all these temporary kind of bondings. We do not check for covalent bonding. It is like that. Okay. Next. So coming to pharmacophore modeling, it is another type of method that is used in computer drug design. It is like, as I have said, molecules acting on the same receptor, the same target, they are having similarity in their structures. So that similarity we need to find out. And as I have said, we want to know about the certain things about the drugs, like hydrogen bonding, electrostatic interaction, lipophilic interaction, and then van der Waals kind of interaction. And all for all these kind of things, we need to have those hydrogen bond donor, hydrogen bond acceptor. These are the certain kind of things that we used to search in the drug molecules. So now suppose you have 100 molecules, active molecules are there, and all 100 molecules you keep one upon other, align the structures one upon other, you will find some common places. Common places will be there where in some common places all the hydrogen bond donors will be there. In some other places hydrogen bond acceptor will be there. And somehow if you link those things as it is shown here with line, you will get one kind of framework. And that framework it is called as pharmacophore. And when we model this framework, it is called as pharmacophore modeling. It is like you take 100, 200, 300 molecules, active molecules with their activity, and you model one pharmacophore, common pharmacophore for them. You get the pharmacophore. So that framework you will get. Now use this framework to screen new molecules. You go to Zinc database, download molecules, millions of molecules you can download. Use this pharmacophore to screen all those molecules. So the software it will give you the molecules, those are matching with the pharmacophore. And it will predict the activity for you even. How much the activity can be. This probable activity. Okay. All the activity, what it is shown in case of in silico studies, these are probable activity. So it will show you the probable activity also. That is about pharmacophore modeling, right? Next. So it is this kind of thing. So you will be getting means hydrogen bond acceptor, hydrogen bond donor, hydrophobic core. So ultimately, you can design one pharmacophore, and that design pharmacophore it can be used to screen new molecules, or even already available drugs. You can screen through this pharmacophore. because nowadays new concept it is coming. It's like not nowadays, means already it is there. It is like drug repurposing. Many examples are there in drug repurposing. Means suppose one drug is there already available in the market for some other kind of activity. The same drug it can be used for another activities also. The best example is aspirin. Aspirin when it is dose is 500 mg, 500 mg dose, it is used as analgesic, means for pain relieving. But same aspirin, if its dose is 75 mg to 150 mg, it is used as anticoagulant drug for heart patients, for coronary patients. It is used as antiplatelet aggregation drug. So that is drug repurposing. So this kind of pharmacophore model can be designed and all the available drugs can be screened. How they are showing the activity, because all the available drugs are what? All has passed through clinical trials. So if you can find out some drugs for other activity, so you need not to go for clinical trials, right? You will have to go for clinical trials, but for toxicity, it has been already screened. So from one side, you will be in plus point. Toxicity is already screened, so the drug is in the market. So same drug, if you can find out its other kind of activity, so it will be very good for you. It is that kind of thing. Again, like even many other drugs are also there. Those are used for 2 3 different kind of activities. Okay, so that kind of things can be done. Next, de novo drug design. De novo drug design means it is like when we design a drug, we try to find out, is there any kind of previously available drug for that particular target? Suppose if now I would like to design a COX-2 related drug, I will go for the structure of paracetamol, aspirin, diclofenac, aceclofenac. Then I will try to design my drug so that it looks like them. Okay. But de novo drug design means it is totally new. Suppose you know the target, you know the protein, you know the active site, but already available drugs are not there. You are totally means designing a new drug. So how to design the molecule then? So you do not have any kind of previous references, previous structures. So now you will have to design the drug. So it is totally done inside the active site. Inside the active site will be checked for its different hydrogen bond donor, acceptor, and different other sites. And inside that, your new molecule will be built up. So that is called as de novo drug design. When a very new drug is designed, where in that particular class you do not have any kind of previously designed examples. We do not have any kind of drug. Totally, we are designing a new drug based on the active site. That is called de novo drug design. So same thing happened in case of COVID also. COVID, we were not having any kind of medicines, no drugs were there. That based on that protein structure, de novo drug design was done. That can be done. So that is that kind of thing it is. Okay. Next. So basically all these computer-aided drug design modules, we can differentiate to two types. It is like target-based drug design and ligand-based drug design. Means when we know the structure of the target, structure of the protein, the enzyme protein or the receptor protein, whatever it is, if we know the structure, then it is called as structure-based drug design or target-based drug design, because we are designing the things based on the target. In case of molecular docking, it is target-based drug design. In case of de novo drug design, it is target-based drug design or structure-based drug design. Similarly, if we do not know about the structure of the target, only we know the previously available ligand molecules. Based on the previously available ligand molecules and their properties, if we design, it is called as ligand-based drug design. It is like in case of QSAR. In case of QSAR, based on the already available ligands, only available ligands were considered, not the target was not considered. So, in case of Pharmaco Modeling, only the available ligands were considered. So, if it is done like that, it is called as ligand-based drug design. So, structure-based drug design means when we consider the structure of the target protein, that is structure-based drug design, and ligand-based means when we do not consider the target structure, we only consider the structure of the ligands, available molecules, that is called as uh, ligand-based drug design. I think this is the last slide, and now the thank you. Now the platform is open for questions.
So, thank you so much, Dr. June, for a very insightful presentation. This was a very clear and informative talk. So, it's time to take some questions now. If you have any questions, please post them in the chat. So, our first question is, in the lock and key method for ligand binding to drug, is there any method to predict the best conformation of a ligand molecule that can bind to the target?
Yeah, definitely means the software will show different conformations and with their binding energy. So the conformation having the maximum negative binding energy, that is the best conformation.
Thank you, sir. Next question, please. The next question is, can ADMET properties of a molecule be predicted with in silico tools?
Yes, yes, different tools are there. Different free tools are there, paid tools are there. You can go for that. For Discovery Studio, we have Topcat, right? So
Yes,
different
sir.
tools are there. They can be easily predicted.
Yes, thank you. Next question, please. If a compound has been identified that has targeted activity and suppresses a disease, but also has toxicity, can the toxic part be modified to non toxic and still used?
Yes. So that is the beauty in drug design. We in one run we cannot get a drug with activity and means less toxicity. Many times it happens we get a drug molecule where activity is very high, but it is showing toxicity also. Then again it is cycle. We come to the initial position, again redesign the drug. We try to find out which are the groups they are giving the toxicity. Then we reduce the group, again we go for the activity. Finally, in this after several cycle like this, we get a drug molecule with activity and less toxicity.
Can we have the next question, please? There is an ethical issue with respect to testing drugs on animals. Are any in silico models available for testing if a drug is toxic?
Yeah, nowadays ethical issues are there to get the means toxicity test done in animals. But if your work is genuine, if your model is fine, then you can go for animal ethical committee. They will permit you to get the toxicity done. We also doing like that only here. We have animal house ethical committee is there. They used to allow us for that animal activities. It is fine. And your question is like, are any in silico models available for testing if a drug is toxic? Yes, many in silico models are there. Softwares are there. Like Topcat is there in case of Discovery Studio. Then other things are like Osiris property calculator. Those things are there. They are free to user. And this can be done. Means toxicity can be screened. in silico. But that is your probable value. Whatever the thing we are doing in silico, these are probability. Okay. Nowadays, success rate it is very high for in silico techniques because systems are upgraded, softwares are upgraded. Okay. So that also you means that is probable thing. After that, you can go for animal studies again, taking the result of in silico. That is how we proceed.
Thank you, sir.
It will be more easy for you
Any
to get the permission then. Yeah. Please.
The next question is, can we patent a drug designed or repurposed with only in silico tools?
No, no, no. This cannot be done. This has to be in silico is preliminary means checking the probability of the things. But the final thing it has to be done in the wet lab only.
Okay. Do we have any more questions? Okay, it looks like we are done with our questions, sir.
Okay.
So, thank you so much, sir, for a very insightful presentation. This was a very clear and informative talk. So, it's now time to felicitate our speaker of the day with a certificate of appreciation.
Thank you. Thank you very much.
So, thank you once again, sir, for giving your time. Thank you for your valuable perspectives that you've given to our audience and for the great interaction. And I'd also like to thank the participants as well. So, if you want more such informative sessions, please subscribe to our channel and please leave a like as well. So, yeah, thank you so much, sir. It was a
Thank you.
pleasure to have you with us today.
Thank you very much.
Thank you.